Hi, good day all, and welcome to UTT's webinar series, Hype or Hope, Investigating the COVID-19 Vaccines. I am your moderator for this evening, Ali Nisas, one of the senior nurses at UTT. I have the great honor of introducing our presenter this evening, Dr. Nicole Ramlachan, Associate Professor in Biotechnology at UTT. Dr. Ramlachan is an expert in the area of genetics. She holds a PhD in genetics from Texas A&M University, USA, and a BSc and MSc from the University of Guelph in Canada. She has extensive experience in the areas of genetics, specifically in the areas of immunogenetics, clinical genetics and genomics, bacterial and viral biotechnology, forensic DNA analyses, and molecular biology. She has spent the last 25 years doing teaching and research in 7 countries, in academia and private companies, as well as for the US and Canadian federal governments and international agencies based in Central and South America. Dr. Ramlachan was also involved in the sequencing of the bovine and cacao genomes and is well published internationally. She has worked with the local ministerial appointed DNA committee, is involved in genetic testing locally, including SARS, COVID-2 testing, and research. She has an extensive array of accolades that I did not mention, and so without further ado, may I present our aptly suited presenter, Dr. Nicole Ramlachan.
Good evening, everyone. Thank you for that lovely introduction, Ms. Nisa. I'm glad to be here this evening to start off this COVID-19 series. We decided we're going to do it in parts because really the COVID-19 pandemic is a story, so to speak, and we're going to do it like in sessions to tell the story. But we've started today with one of the more concerning areas, which are the variants. So I'm going to share my screen now, and hopefully we will be able to start with that. So introducing the webinar series. It's called the UTT COVID-19 webinar, Hype or Hope, Investigating the COVID-19 Vaccines. And what we intend to do is to try to delineate some of the issues that we may be having with people who are trying to figure out exactly how they fit into this whole scene with this particular virus. So the first session we're going to talk about is COVID-19 and variants, the emerging threats, in terms of whether or not these new variants that we're seeing are really as dangerous as we think they are. But before we start that story, I have to give you a little biology lecture now. I go into professor mode, and to just to kind of introduce the topic of genetics, because this is all about genetics really at the end of the day, how we identify the variants, how we have identified COVID-19 causing virus SARS-CoV-2 to begin with. So genetics is kind of a complicated kind of story because there's a lot that's lost in the translation, right? So these people similarly were trying to translate their language into English and failed. And that happens a lot with genetics as well. But what exactly is a gene? So a gene is what encodes who an organism is, whether you are human, a virus, a sheep, lizard, bird, whatever it may be. And that is encoded in your cells, or in the case of single cell organisms, in the cell. And if that is mutated, it causes changes. In humans and animals, we can get disease because of those mutations. In things like viruses and bacteria, it gives them certain powers, sometimes superpowers, to be able to do things that they would not have been able to do before. And that nucleic acid allows us to be able to identify different organisms and say, okay, well, this is this, or this is that, or whatever. So initially, when COVID-19 pandemic, or previous to the pandemic, when they identified the SARS-CoV-2 virus, we realized very quickly that it came from the family of the SARS. And we had seen SARS before, back in the 2000s and 1990s, when there was the original SARS pandemic. And that was also caused because of a zoonotic jump from bat to human. Similarly, with the SARS-CoV-2, we know that it was a zoonotic jump. So a zoonotic jump means that it's jumped, a virus or disease has jumped from an animal source or animal host source to a human. In the case of our particular pandemic, COVID-19 causing SARS-CoV-2, we knew it came from bats because of the sequence data that we were able to look at. They may have been intermediate hosts like pangolin and camels and those kinds of things, but most immediately before our infections, it came initially from bats. And how did that occur? Well, basically because as humans, we encroach on the habitats of these wildlife. We just have a lot of habitat destruction as well too, and wildlife trafficking, illegal wildlife trafficking that causes that, and they get exposed to humans, and the viruses love nothing better than a different host, because viruses are just single cell organisms that are trying to survive. Things like Ebola and bird flu and swine flu and stuff we've seen before too that have been able to jump from animal host into a human host as well in the past. And how do they do that? Well, let's go back to the actual infection basics. So this is a little diagram here of the SARS-CoV virus, CoV-2, and these black things that's sticking out are the spike proteins. And these are the important artillery that the virus has that allows our cell receptors to be fooled into allowing them in. So they've been able to mutate just enough away from the bat or whatever the last host was, so that it changed that particular spike protein so that all of a sudden now our receptors are able to recognize it and let it in. And when we let it in, our cells now get fooled into being that virus's a manufacturer. So our cells just, specifically our alveolar cells in our lungs, will just now spend all its time until it is lysed by the virus as well too eventually, or dies naturally, producing viral proteins and assembling them and then spitting them back out to go off and infect other cells. And then eventually with COVID-19, it will infect immune cells, and which will in turn cause a big problem in the body. So this is kind of what happens at the end of infection. We can have all kinds of different symptoms, and people can have different degrees of severity of disease. Eventually, some people can have absolutely no symptoms, be totally asymptomatic, or be able to just have fluids, rest, and acetaminophen, all the way, of course, to ventilating because of respiratory failure, dialysis because of kidney failure, and then eventually death, or if they're able to recover, discharge with usually long-term, long-haul symptoms, we call it, right? So SARS-CoV-2, of course, all around the world, this is today's data, about 147,200,000 cases, and deaths are right around 3 million, right? So it's been increasing lately. We've been in a spike basically worldwide. Trinidad and Tobago, you'll see right here, has 9,676 cases as of today. And in terms of average daily cases, we're at about 140, which is upwards in the 14-day trend. You can look at it here. And everything that's really dark red is increasing and pretty high in terms of caseload. And the ones that are yellow, of course, are not as high. We've already passed 147 million cases, but what has happened is that it just took 1 month to go from 80 million to 100 million, whereas it took almost 2 months to go from 20 to 40. So that means the rate of infection is increasing, which you can see if you look at all the different regional comparisons that we've looked at here, including the individual countries in Latin America and the Caribbean. Everybody's kind of ending in an up spike, or upturn, which means that cases are increasing right now. If you look at the deaths per 100,000 people, I just included a couple of other countries in this slide here. Trinidad and Tobago is down here. The darker black and darker red, the more number of cases, and the lighter yellow, of course, down to white is 0. Initially, you can start to see everybody else started off at 0 except the US and the UK, and then eventually we were getting into the darker colors for Turkey and Tunisia in particular. We are right at about 11.4 in our death rate and about 159 deaths as of today. And a lot of this data comes from epidemiological data as well too. So now what do we know about the SARS-CoV genome? The genes contain different parts to encode all those different parts of the virus I showed you earlier, right? And here we're actually looking at the SARS-CoV genome as a linear format, right? So here's the open reading frame, 1, protein 1, and spike protein, and then a bunch of other proteins down here. The spike protein is the one I showed you, and that's the one that we're most concerned with in terms of how the virus gets in and out of the human body. And the genetic instructions in the brown part is where the region that we spend a lot of our time sequencing to be able to monitor the mutations. So what exactly is a mutation? I mentioned before, it's a change that causes a sequence change in an organism's DNA or RNA. In the case of COVID-19, the variant mutations are on the mRNA because they are mRNA molecules. And a new SARS-CoV variant has one or more mutations that differentiates it from the predominant virus. So initially, when they started off, this was the main virus over here, the D614G, which came out of China, and then it mutated and became variants of concern. Three main ones: the B117, which we call the UK variant, the B1351, which is the South African variant, and the P1, which is the Brazilian variant. And if you notice, it's not one mutation. The United Kent variation has 18 mutations, which differs it from the original Chinese virus that was isolated. The South African has 8, and the Brazilian has 21. And the mutation specifically in the S region of the spike protein, these are the main ones here, right? And this causes possible functional changes that we've seen. They seem to be able to be more efficiently transmitted from person to person. It may have reduced antibody binding and immune protection, but that's not confirmed to date. And it may reduce vaccine efficacy, specifically with the B1351 and the P1. Again, which we haven't been able to test with all vaccines. Some vaccines, which I'll talk about after, were exposed to that, right? But basically, we have to constantly do surveillance and do sequencing to look at how some of these variants can impact the disease, transmission, or severity and effectiveness of vaccines and therapeutics, right? So that's what we know. Has this happened before? Has the SARS mutated before? Yes. So initially, it started off as the D614 SARS-CoV-2, if you notice, from January 11th. And then all of a sudden, you started seeing this G614 appearing, to eventually the G614 was dominating the scene. And the G614 had a magnitude of infection that was much greater. So I don't know if you all remember that we initially started talking about outbreaks and stuff in China and South Korea. And then when it hit Europe, it started to get really, really bad in Italy and those countries, and Belgium and Netherlands and so on. And they were the ones that were suffering from influx of this particular variant. So this is, again, graphically here, geographically, I should say. Prior to March 1st on the left, you were seeing the blue is the G614, the yellow is the D614. It was not really predominant, and as of March 21st to the 30th in 2010, it was. So this was the last year. Of course, since then, now we've been able to do a lot more sequencing and get a lot more data and do a lot more analysis. And we know now that there are 3 types of variants. You have a variant of concern, variant of interest, and a variant of high consequences. Some variants have been already classified as variants of interest. Others are variants of concern. To date, no variants of high consequence have been identified in the world, which is a good thing, because that basically means that nothing that we have would work against it. And then why does this happen? Well, basically because the virus, of course, are changing and mutating. And how does it become a variant of concern? Well, there are 4 things that we're concerned about. If it affects the transmissibility, the virulence, as in the severity of the disease, vaccine effectiveness, or diagnostic testing, right? So these are the variants of concern that we know about: the B117, P1, B1351, B1427, and the B1429. And here's where they were first detected. And so far, what we know about them is that they have increased transmission, most of them, from 20 to 50%. They have modest decrease in terms of the antibody treatment. So a lot of the treatments that we have don't work well against the variants. And that's why you're seeing a lot of severe disease and death associated with these variants, because they are infected for longer, so they're transmitting faster, and they are staying sicker longer, because we aren't able to actually get their symptoms under control fast enough, right? This is the P1 variant that we hear about all the time, that's now in Trinidad, or otherwise known as the Brazilian variant, and it has these, everything where you see a red point is a mutation. So this one has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 mutations, right? The variant of interest is a little step down from that, where there's specific genetic markers that have been associated with changes to receptor binding, and it is able to reduce the neutralization of the antibodies generated against previous infection or vaccination, or reduce efficacy of the treatment's potential diagnostic impact or predicted increase in transmissibility or disease severity, right? The possible attributes of the variant of interest, they have specific genetic markers that are predicted to affect transmission, diagnostics, therapeutics, or immune escape. This is a scary thing that we're concerned about, that it's not able to respond to vaccines. And is able to escape immunity, right? There's evidence as well too that if these variants of interest cause increased proportion of cases, that's of concern or limited prevalence or expansion. And these are some of the examples. But as I said, they tend to coexist okay with the normal variant because we're able to actually fight them effectively. They may have a little bit of reduction and neutralization of some of the antibody treatments and so on, but generally people are able to respond okay, right? Any variants of concern, a variant for which there's an evidence of an increase of transmissibility, more severe disease, increased hospitalizations or death, reduced reduction in neutralization by antibodies generated during previous infection or vaccination, reduced effectiveness of treatment or vaccines, or diagnostic detection failures. And these are all the impacts, right? Widespread interference with diagnostic targets, substantially decreased susceptibility for therapies and neutralization of vaccines and those kinds of things. And basically, we are worried about the vaccine-induced reduction in severe disease. This is just to show you how it trends. As I mentioned before, viruses mutate all the time. We have no control over their ability to mutate, and they are survivors, and they've been here billions of years ago, and they'll be here millions of years after we leave, and they are going to mutate. If we look at the mutations in the US of the B12, B1596, and B1 others, and then here's B1429 and B1427, and the prevalence, you would notice that it changes over time, right? So we started off with mostly light blue, now we're mostly dark yellow, right? And everybody else has changed over time. And this is here just the most common lineages and percentage. So basically in our parts of the world, B117 is the most popular in terms of variant that we've been able to sequences. So what are the steps that we do? Well, basically a variant of concern, what can we do? They can basically say, okay, well, we have to increase testing, control spread, start to put in lockdowns and those kinds of things, which has happened to us here in Trinidad and other parts of the world, where we're able to actually say, okay, well, we need to control spread. And the way we control spread is to control exposure, right? So that's one thing they do. And then of course they ramp up testing and all of those things and sequencing and those things as well too. And then the range of interest, of course, you would do more things like just doing more surveillance, right? So far we have the B117, which is verified. We've had some cases of the P1 reported as well recently. That is not verified so far on the international front, so that wouldn't come up on that graph. India has been in a state of despair over the last few weeks. They have a 7-day rolling average that is basically an exponential straight line right now for numbers of cases. That's related to basically non-compliance to mask wearing and reduction of vaccination programs as well too, because they're only at about 10%, a little bit less than 10% right now, their population vaccinated. And a lot of areas that have gone through a lot of religious festivals and things like that, and they've said that a lot of that has come to it because of exposure, and their government is not locking down or limiting movement across the country. Makes it very difficult to control. Toronto, recently there's reports every day of their lockdown. They're actually back down in a lockdown right now because their COVID-19 variants are actually making people so sick so fast that they're actually dying at home and not even getting to the hospital. And there are some cases of some children dying as well too, which is quite distressing. So how did the variants occur? Well, basically, of course, that genome that I showed you can mutate, right? And what we're doing is we're monitoring the circulating viruses for the key mutations that happen. Now, many of these mutations happen naturally and nothing happens. The virus moves around normally, and there's no change in how it spreads or causes disease because it doesn't alter the major proteins involved in infection. But sometimes they become outcompeted by variants with mutations that are beneficial for the virus. And then that affects us because it becomes more infective. Are all variants bad? No. Most of them won't persist or change the nature of the virus. If millions of people or hosts are exposed, if we are unvaccinated and we become carriers and we become hosts, that's where the problem happens, because then the mutations occur much faster and then they can actually be able to be allowed to transmit much better cause more infection, cause more severity of disease, and possibly evade our immune system, which is a big no-no we don't want, right? We might have to eventually have to boost vaccines. So you'll hear the vaccine manufacturers talk about boosters, even though we've gotten two-phase orders. We might have to do another booster. I think Pfizer's already said that they want to do a third booster. Or change the vaccine makeup so it's more universal, so it contributes to protection against all the variants. And we constantly sequence the new variants. So this is, I just wanted to kind of show you all, this data is real. Here's a graph of or a key of all the variants, all the colors that was seen, and here's where they were isolated. And this is how they are related. So we're able to actually interpret these phylogenetic relationships doing these kinds of genomic sequencing projects. All this data is online. Everything is publicly published. It's very easy to see and get and download, and for us to be able to worldwide be able to contribute sequences, get sequences, and be able to monitor those changes, and be able to use really good surveillance methods to say, okay, well, this variant has come up here, and so on. What about the vaccines and the variants? So the COVID-19 vaccines so far have shown to produce some protection. The changes and mutations in the virus should not make the vaccines completely ineffective, as we know today. If they prove to be less effective, it's very easy to change the composition because most of them are very easily changeable in terms of the starting production of vaccines, which we'll talk about later on in the series. Are they protective? Yes. Well, basically what we did was we took the first original COVID-19, SARS-CoV-2, and we created a vaccine for it over a year ago. By the time the vaccine came out, we had about 4 variants. We were lucky in that AstraZeneca and a couple of the others were tested out in South Africa and Brazil, and in particular in Brazil, where they had the P1 variant. So we know it's effective against those variants. Some of the earlier ones like Moderna and Pfizer, we don't know to date, and the studies are ongoing. And of course, some of the other ones like Sputnik and the Chinese ones and so on, again, we have some data to show that it's protective, but that data is ongoing. So we might eventually have what we call a waning immune response, and the variants might emerge later as they will be able to outcompete. So that's one worry that we have. Basically, what we're doing right now is studying all of the current coronavirus vaccines constantly. Remember, this situation happened very quickly. We were able to produce a vaccine in record time, in less than a year, which has never been done before. And it's because of the amount of technology that was available to us, and of course, the amount of money that was spent by many, many countries around the world to be able to do that. But still, there are going to be lots of ongoing research to be able to see how effective they are against the mutated variants, right? The Ken variant has shown to be effectively prevented against the AstraZeneca Oxford. And there's less evidence, as I mentioned, about the Brazil and South African, but we know AstraZeneca works on the South African in particular, and now the Brazilian because of the testing there. And they were able to actually do sequencing to be able to find those variants initially and be able to see how it is that they actually mutated and how they can actually, vaccines can actually prevent against that particular transmission of disease. Are they more effective? Well, it's difficult to assess because right now we have a lot of vaccinated old folk. So if we're seeing a lot of young folk that are sick, it's maybe because they're not vaccinated, or it may be because the new variant is able to infect them faster. So it's a kind of a catch-22 situation when we're looking at those studies. But evidence is building that it seems to be causing higher infection rates in younger people, and the unvaccinated children can still transmit to each other and older people, even though they do not show high rates even today. There is a need to accelerate the vaccines to include the youth and children as well too. What about traveling with the variants? Well, globally, the policy has been to restrict travel on countries with the different variants and the high outbreaks. And the variants of concern in particular have been breaking through immunity and causing disease are very concerning because we're still vaccinating. No country has reached really high in a percentage of 2 vaccines yet, except I think Israel is pretty up there, and Bahrain and some smaller countries like the Cayman Islands and so on is at 90%. We have to be able to reach some level of herd immunity to be able to evade that, right? And we have to balance the policies of lockdown and lock-ins, of course, with the socio-economical costs. We were trying to do this to achieve herd immunity, and herd immunity basically is the indirect protection of infectious disease that happens when the population is immune through vaccination or previous infection. Now, we know previous infection does not cover individuals because they've been able to be reinfected in the past. So we are looking at a vaccination for everyone, even individuals who are recovering, or long haulers and those kinds of individuals as well too. It will be achieved when 95% of the population is immune, either through vaccination or through natural immunity. And we have to get there, right? And the only way to get there is and shrink this death rate that is increasing daily all over the world is through vaccination. So we have to stop the virus in order to be able to get less variants. The way we get more variants is because the virus is in the population circulating. Manufacturers might have to tweak them to change to the evolution of the virus and the variants as much as we can. And they might have to incorporate more than one strain or have booster shots or make other kinds of changes. And that will come in time. But basically, we have to get to the point where we are vaccinating as many people as possible to reduce risks, right? So I just want to leave that with you. The way that we treat with variants, get vaccinated. The answer is always get vaccinated. Okay, thanks very much for your time.
Okay, thank you, Dr. Nicole Ramlachan. So first off, we have one question so far by Marcus. Are we at risk for facing a situation like India?
Well, that's a hard question to answer in that we are not to the point of being vaccinated at any percentage that provides any level of herd immunity. We are at 1.2 something, barely 1.3% as of today. I think we actually, let's round it down because it's 1.2. We're 1.2% as of today. India has 10%, and their situation is very, very bad. Of course, their population is very great. So even though they've given 144 million doses of vaccine as of today, that is a drop in the bucket. We have given about 36,000 as of today, I think. And again, a drop in the bucket. So we have to ramp up. We are at risk in that we don't have the facilities, the ICU beds, the amount that we would need to be able to combat anything of that kind in terms of the rate of infection. And that is where transmission becomes an issue. And we must take precautions. People are suffering from what we call pandemic lull, I guess. I don't know. People calling it different ways and different things. And they want it to be over. And so they're dropping their guard. And we can't have that. We don't have the vaccination rates to be able to cover that, and we won't probably have it for another year or so. The variants are there, and they're going to continue to mutate, and they're going to continue to become present in our porous borders and spread throughout the country. So there's always a risk for us getting into a worst case that we are right now, I would say, and the only way to end that is through vaccination.
Okay, all right, thank you. So I want to invite everyone, you may ask your questions via the YouTube chat box. Dr. Ramlachan, I just wanted to ask one question as we wait for the questions to come in. What are some of the long-term symptoms post-discharge?
Sorry, I didn't hear you. Just repeat, please.
Okay, well, I will come back to that. We have a question by Dr. Varma Rambaran. There has been a popular video circulating on social media from a top member of Pfizer stating that the vaccine will suppress natural immunity against other viruses. Your comments?
I don't know who that is. Haven't heard it, haven't seen any publications on that. Nothing so far that has been published on any vaccines shows suppression of immunity to date of any of the papers that I've read. So I can't really comment on that in particular, other than to say that there's a lot of videos, a lot of people self-reporting experts on every single thing. And so you have to take all this data with a grain of salt and perhaps go and to the peer-reviewed original articles and be guided by that. The main thing you have to be guided by in terms of your vaccination is that as of today, we've vaccinated almost 500,000 people in the world. So, and they're all walking around mostly normal. Some people have had reactions ranging from shoulder pain to, of course, hospitalization in the case of the people with the blood clots, but they have been in the tens, not even the hundreds. So, and in terms of long-term effects of the vaccine, obviously we won't know. because we haven't been able to do long-term studies, except of course with the phase 1, phase 2 trials. So in these cases, the long-term is 10 months, 11 months. That's the maximum amount of data we've had, and nothing looks awry so far to date in terms of immunity.
Okay, great. Mark asks, with the possibility of Brazilian variant in the area and available vaccines may not be as effective, what is the advice on which vaccine to take?
The AstraZeneca has been shown to be effective against the Brazilian variant, and that's what we have here locally.
Okay, I asked earlier, what are some of the long-term symptoms post-discharge? So you were infected, you recovered. What are some of the symptoms that you mentioned that may occur post-discharge later on in life?
I think we're going to cover that later on. It's nicer in the long haulers. We have a whole session on long haulers and those kinds of individuals, but it ranges from headaches to kind of like immune disease kind of disorders. So a lot of joint pain, leg pain, aches, interesting things. There's something called COVID toe, where your skin peels off your toe. Then of course there are the people with the serious side effects, which are or long-term effects, which are heart-related, digestive related, and kidney and liver related, and those are much more serious because they had damage. And of course, the people with respiratory disease that lasts long term because it's a respiratory, it's first and foremost a respiratory virus.
Okay. Okay, so your persons are, okay. So again, I recommend persons can ask their question via the YouTube chat box. You did mention about the surveillance of variants. How prepared is TNT? This is again my question. How prepared, in your point of view, is TNT to provide such surveillance?
Well, they're limited, obviously, because surveillance will come with sequencing. So there's only a few people who are involved in that research-wise. But in terms of the testing, anybody can do it. In terms of the antigen testing and those things as well, too, as those kits become more available, normal labs are doing it, and eventually they're going to get to the point of having home kits where you can do your own antigen testing at home eventually. And that's probably where it's going to get to. Because the RT-PCR testing is, of course, costly, more complicated. You have to go through the public system, wait a couple of days, those kinds of things. And that kind of surveillance becomes a little moot when we want to know whether or not somebody is sick quickly. And that's where the rapid kits come into play. So those kinds of rapid tests are going to take over soon in terms of how we are able to use surveillance. Because as you know, surveillance is something you need to know as fast as you can, right? Because you need to be able to do contact tracing and all of that and isolate that individual quickly. And the way to do that would probably be to move to more rapid kits.
Okay,
which we have here, by the way, already.
Okay. Saeed asks, do we have an estimated timeline for how long it is going to take to fully vaccinate our population?
When you say estimated timeline, do you mean that what they are doing or what they should do? Because that's two different things. So what they're doing right now is going to vaccinate us if it is that we have no pause, which we probably have right now, because we are almost out of the first batch of vaccines, waiting for the second tranche of COVAX to come in. And I think Minister Yelsing was estimating 1,000 per day. If we move at 1,000 per day, we will be vaccinated sometime in 2024. So that's the rate at which herd immunity will be achieved if it is we proceed along the way that we are doing. Clearly we can't, right? Because nobody can wait till 2024 to, play carnival and play mass and do business, right? And open companies and, do business like normal. It's going to not be feasible. So we have to ramp up.
Okay. Another question from Kiran. What would you say to the individuals who want to wait for the Moderna or Pfizer vaccines because they fear or don't trust the AstraZeneca vaccine due to blood clots? Will we ever get those vaccines?
Well, funny you should ask, Mr. Kieran, because we're doing that on our next topic on Friday. We're talking about exactly that, the efficacy of vaccines and whether or not it makes sense to wait for one over the other. The general recommendation is take whatever vaccine you're offered. That's what every country's recommendation is, including the WHO, which is the overall governing body, and the or directing body, I should say, and all the individual people who are involved in the vaccine distribution. Any vaccine at this point is going to protect you 100% against severe disease and death. And that's what you want to do.
Okay, we have a comment from Martin. Great info, powerfully delivered. And also a question, are there any dangers in a family of 5 having 3 or possibly more different vaccines?
I don't know. I mean, danger as into people who've had the vaccines. No, there's absolutely no exposure to your immune response inside your body to a vaccine you've taken to someone else. There's no effect. So the immunity that you get or any reaction that you get from an injection, which is a vaccine containing injection, will be your own. You can't affect other living organisms with your immunity.
Okay, all right, thank you.
The danger might come in, I just want to put a cut. The danger might come in individuals who are eligible for vaccines. So right now we're not vaccinating children or anybody under 18. Well, actually in Trinidad, we're not vaccinating anybody under 60 technically, who's not a health worker, who has a comorbidity. So it becomes a concern when people are vaccinated in the household and other people are not. And then they can be transmitters of the virus, not show symptoms, and bring it home. So we have to be very careful, even between vaccines and within families where there are several vaccination profiles, that we still observe normal protocols for reduction.
Another question, John asks, is it safe for HIV persons?
Yeah, the data showed early on that there's no issue with individuals who are HIV. They did a phase 1, phase 2, 3 trials with individuals who were infected with other viruses in addition to individuals who had cardiovascular disease, diabetes, obesity, those kinds of things, because those are the individuals who were susceptible to COVID initially. And yes, there doesn't seem to be any known particular threat to individuals who have HIV in particular that I've read.
Okay, so no more questions thus far, still waiting. So everyone, you can send your questions via the YouTube chat box, right? Any closing comments or statements, Dr. Ramnacha?
I just wanted to say thanks for this opportunity. I'm very happy that we're able to do this and reach out to the public. I like the idea that we're able to do it in short snippets as well too, so that we're able to maintain interest. And thank you all for being so attentive and asking such great questions. I appreciate it. And I hope to see you all on for the other 4 sessions that we have, because it's going to be very informative. And I think we'd be able to answer a lot more of your questions as we get more into the meatier details of exactly how COVID-19 vaccination is going to end this pandemic and basically save the world at this point.
You want to give us a little tidbit on what your presentation will be on Friday, just to wet the throats of the listeners?
COVID-19 vaccination, yeah. So it's going to be exactly that, the actual vaccination and vaccine story.
Okay, great. So thank you, everyone, and have a great and safe evening. Thank you, Dr. Ramnachan, and that is it. All be well.
And Friday, I think we're on at the same time, I believe, right? Same time, same link. So